Welcome to our video lecture on proteins and enzymes. There's going to be a lot of information here, super fast, but we're going to practice it in class, so don't panic if you don't catch everything right now. Our essential understanding for proteins is that they have a very wide range of functions in living organisms, and these proteins on the screen right here are just some examples of the many proteins that we have in living things that help us do work and hold us together. Lots of objectives for today. We're going to understand that proteins are chains of amino acids held together by peptide bonds. That's an easy review from 9th grade bio. We're going to look at amino acids that have different properties and how those properties are conveyed to the proteins. We're going to talk about those 4 levels of protein structure: primary, secondary, tertiary, and quaternary. We're going to talk about how proteins can be globular or fibrous and how they can be denatured, and that the function of a protein is related to its structure. And then we're going to flip over to the idea of enzymes. Enzymes are proteins. The function of them is metabolism. We're going to look at the active sites of enzymes and how enzyme activity rate can be altered by lots of different factors. And then we're going to talk about how enzymes can be used in technology. So up first is amino acids are linked together by condensation reactions to form polypeptides. We know this already. We've been doing this in class quite a bit. Make sure that you can draw a couple amino acids getting squished together by a condensation reaction to form a dipeptide. Peptides means protein. A dipeptide is 2 amino acids, 1 and 2, stuck together with a peptide bond. Polypeptide is lots of amino acids stuck together with peptide bonds. And we'll talk more about translation later, but the formation of this peptide bond, that peptide bond right there, is done by ribosomes. So this is the same slide as the previous slide, except the picture's not here because we are going to draw it ourselves. Remember that we're going to start with that central carbon. We're going to put an amino group on that central carbon, N and 2 H's. That central carbon gets a hydrogen and a radical side group. And then we're going to have our carboxyl or carboxylic acid group over here. Because we're making a dipeptide, we need to show our second amino acid. So again, central carbon, amino group. You should be drawing this with me. Hydrogen, side group, carboxylic acid group. Good. So now what's going to happen is we're going to take the OH of one carboxylic acid group and the H of an amino group, and they are going to combine to form water, which is why we call this a condensation reaction. What remains is a dipeptide, because the bond that just got broken right here and the bond that just got broken right here, these guys are going to find each other and we're going to make that peptide bond. What that will look like is this. We'll still have our amino group on this end and our central carbon with an H and an R. We'll have that carboxylic acid group, but it's not a carboxylic acid group anymore because the OH is gone. So now I just have that double bonded O, and then that carbon is going to go straight over now to the nitrogen of the amino group of that second amino acid. It still has one of the hydrogens, but only one, not 2. And then again, central carbon, and on this end, we do still keep the whole entire carboxylic acid group. That carboxyl group is there. Label that peptide bond. This whole thing is a dipeptide. Here we have one amino acid and our second amino acid. Amino group. Carboxyl or carboxylic acid group. Good? Good. Make sure that you can do that yourself. So when we combine these amino acids into polypeptides, crazy long chains of amino acids, we get a variety of proteins, and some can be quite small and some can be very large. Insulin is an example of a fairly small polypeptide. It's composed of only 2 chains of amino acids. One is 21 amino acids long and the other is 30. So insulin altogether is only 51 amino acids. Titin, which is this super cool protein in muscle fibers, is, oh my goodness, 34,350 amino acids long-ish. There's some variations, but it's big. It's a big guy. These proteins are put together by these amino acids, and there are 20-ish. There are some discussions regarding the exact number of different kinds of amino acids, but we have 20-ish different amino acids that we use to build proteins. And they are, we've talked about this before, they are exactly the same except for that side group, that R group. And R groups are going to provide the specific characteristics of each amino acid. Now, I took our generalized amino acid and I flipped it around a little bit because this is how we're going to see it in the graphic over here. So carboxyl group is here, carboxyl group is here, amino group sticking over here, central carbons right here, and then this is the R group. And this is another R group, and this is yet a different R group. So these guys are all positively charged, these guys are negatively charged, and those charges are going to provide different kinds of characteristics to the proteins that have these amino acids. All of these guys are uncharged. These guys are special. Sulfur and cysteine is super, super special. And then we have hydrophobic side chains, and we'll talk about where those guys end up in membrane proteins right now. So again, these different R groups are going to provide some different characteristics to each amino acid. We're going to have some that are non-polar, like this guy, just a bunch of carbons. We're going to have some that are quite polar, like these guys that have hydroxyl groups on them. And what's super cool is when we put them, especially in our membrane proteins, where we remember that the phosphate heads are hydrophilic. They can be attracted to water. And then the fatty acid tails are hydrophobic. They are repelled by water. And so if I have especially like a channel protein, the amino acids that make the outside of the channel that are embedded in the inside of the membrane are also going to be hydrophobic so that they can hang out next to those fatty acid chains. But then the amino acids that are up here are going to be hydrophilic so that they can hang out next to the phosphate heads. And then here's the coolest thing. Generally, the amino acids that make up the inside of the channel will also be hydrophilic, so things like sodium or potassium can get through. Sodium and potassium with those charges are quite hydrophilic, very, very polar. So polar they're ionic, and they're going to be repelled by non-polar amino acids. So we're going to keep the non-polar amino acids out here, polar amino acids on the inside and tops and bottoms. So let's practice that with a bigger picture. It's a little bit easier to see. So in this picture, label where we're going to find hydrophilic regions and hydrophobic regions. Phosphate heads are always going to be hydrophilic. They are attracted to water. Fatty acid chains, the fatty acid chains that are here are hydrophobic. They're going to be repelled by the water. The amino acids that make up this part of this channel protein, they're going to also be hydrophobic because they have to hang out next to the hydrophobic fatty acid tails of our phospholipids. But because I want to send polar things like sodium and potassium or calcium through my channel protein, the inside needs to be hydrophilic so that these things can pass through. So philic phosphate heads, philic on the inside of the channel so that those ions can pass through, phobic fatty acid tails, and phobic outside of the channel protein because that's what's next to that those fatty acid chains. All right, so we have these 20 different amino acids that make all of the proteins for every living thing on Earth, almost and about. And so we have some theories on why. Why are there only 20? Why don't we have more? And it might be that those were the ones that were produced on Earth before life evolved, and so those were the ones that got used. It might be that these ones are the best amino acids, the easiest ones to put together during protein synthesis, and so maybe these were the ones that organisms kept and the other ones just kind of went away. Or it might be that the common ancestor of living things used those 20 amino acids, and so those were the ones that the descendants of that common ancestor continued to use. We don't know. maybe a little bit of both. All three. I can count. It's all good. So thinking about that protein diversity, if I only have 20 amino acids and we make a lot of different kinds of proteins, how is this possible? We're going to do a little bit of math, some fundamental counting principles to just take a little look at what numbers are possible. So if I have my 20-ish different amino acids, how many possible dipeptides are there? Well, if I have a dipeptide, it's going to be two possible things. I have 20 possible amino acids for the first and 20 for the second. That's going to be about 400 different dipeptides possible. And now I didn't account for like the repeats that can go backwards and forwards the same way. But about 400 different dipeptides, just looking at combinations of two amino acids. If I now talk about 3 tripeptides, there are 20 possible amino acids for each of those pieces. Now I'm going to use my calculator for this one. 20 times 20 times 20. It's going to be 8,000 different tripeptides. So thinking about what if I have polypeptides composed of 10 different amino acids, that's going to be 20 to the 10th power. I don't know if my calculator will even do that. 20 raised to the 10th power is going to be 1.024 times 10 to the 13th power. That's a lot of different amino acid combinations, just when I'm looking at 10. And remember that we were looking at Titan is 34,000, so 20 to the 34,000th power. I don't think my calculator is going to do that for me. But the idea here is that because we have these different amino acids in different combinations, we have a huge number of polypeptides possible. So all these different proteins that we make are coded for by our genes. We make about 18,000 different proteins as humans, and the combination of all of the proteins made by a cell or an organism or a group of tissues is known as a proteome. So proteome is all of the proteins made by a system. So about 10,000 of those are present in all human cells, regardless of what differentiation happened. And those other 8,000 are going to be specific to specialized cells. And then all of this information for making proteins is stored in genes, and we call this the open reading frame. So this is the part of the DNA that we actually use to code for proteins. This is the DNA that goes through transcription and then translation actually turns into proteins. What's crazy sauce is that's only about 1% of all of our DNA. Only about 1% of all of our DNA actually turns into these 18,000 proteins that we make. And when we get to our genetics unit, we'll talk about some of the other things that the other 99% of human DNA does. All right, so the proteome, as I said, is that combination of proteins made by a cell or a tissue or a whole organism. And what's crazy cool is that it is unique to each entire individual. Even identical twins are going to have different groups of proteins made. We can isolate and then separate all these proteins using this thing called gel electrophoresis, which is just going to separate out the proteins by size, which is pretty cool. What's amazing is our proteins are actually more variable than our genomes. So we have more different kinds of proteins than we have genes. And it's because different cells make different proteins. And then us at different stages in our life cycles, we will make different proteins. So protein versus polypeptide. Sometimes we use these words fairly interchangeably, but generally the better differentiation between the two is that a polypeptide is just a chain of amino acids, whereas the protein it's actually doing a job. So polypeptide is just going to be amino acid, amino acid, amino acid, amino acid. That protein is going to be, now we've folded it up into some structure and it can do its job. Some of our proteins are composed of only one polypeptide chain, like this enzyme, lysozyme. Some of our proteins are composed of several polypeptide chains that are held together. Hemoglobin, the protein hemoglobin, which carries oxygen around the blood on our red blood cells, as an example, that actually is composed of 4 different polypeptide chains. So I have 1, 2, 3, 4. 4 different polypeptide chains are held together to make this functional protein. So Conformation of a protein, its three-dimensional shape, is going to be determined by the sequence of the amino acids, and then that shape is super important for its job. So these are just some super common proteins, good ones to know: rubisco, insulin, and rhodopsin. Ooh, that's a P. I got cut off by the picture. So these three guys are what we call globular proteins, and we'll talk a little bit more about them in a couple more slides, but these are going to be more metabolic in nature. Metabolic. Collagen and spider silk, we'll talk about these more later. These are fibrous proteins, and they're going to be more structural in nature. We can see that there's a lot of variation amongst all these different protein shapes, and how that protein gets a shape is through 4 different levels of structure. These 4 levels of structure start with primary structure. Primary structure is simply the amino acid sequence. It is the sequence of amino acids from start to end of that polypeptide chain. Secondary structure, this is held together by hydrogen bonding. Remember that hydrogen bonding is because of one thing is a little bit negatively charged and one thing is a little bit positively charged, and so they get stuck together. Electrostatic forces of attraction, hydrogen bonding. And so all of these little dotted lines, these are showing hydrogen bonding. We have 2 kinds of secondary structure. Secondary structure can be either alpha helix or beta pleated sheet. So beta pleated sheet, alpha helix, these are the 2 kinds of secondary structure that proteins can have. All of this is held together only by hydrogen bonding, and those are going to form between different locations in the polypeptide chain. So we're either going kind of this way or we're going kind of this way. And then these guys will like kind of fold around like that, which is why we call it a pleated sheet. Tertiary structure, so the 3rd level of protein structure. Now we have that 3-dimensional folding. It's also going to be held together by hydrogen bonding, but I'm going to add here also covalent bonding and ionic bonding. So we have more kinds of bonds holding together this 3-dimensional folding. There's one super cool special kind of covalent bond called a disulfide bridge. And this is where, I don't know if you remember those couple slides ago where we looked at those different amino acids, cysteine. The amino acid cysteine has sulfur in it. And so if I put together 2 cysteines, I'm going to get 2 sulfurs stuck together, and that's going to be our disulfide bridge. Quaternary structure, this is where we have multiple polypeptide chains come together. So I might have one of these guys and then another one of these guys, and they get stuck together with more hydrogen bonds and more covalent bonds and more ionic bonds. And then those multiple polypeptide chains that are already folded up are going to stick together to make that quaternary, that 4th level of structure. Everything has primary and secondary structure. Proteins are going to have more or less tertiary structure depending on their shape and their job. Not every protein has quaternary structure. All right, so I mentioned a couple slides ago this globular versus fibrous. Globular proteins are literally like globs. They're kind of blobby. They have a lot of folding, and what's super cool is that they're hydrophilic. Amino acids will be on the outside of that folding, and so they are water soluble. The hydrophobic amino acids will be on the inside, kept away from the water. These guys tend to be more metabolic in function. They're often going to be our enzymes. Rubisco is an enzyme, or they might be hormones like insulin. And then rhodopsin is a sensory receptor molecule. It's going to notice some color stuff in our eyeballs. So they can also be receptors, sensory receptors. Fibrous proteins, a little bit different. They are long and stringy, literally like fibers. So collagen and spider silk are both examples. They do not dissolve well in water. They're insoluble in water. They have a lot less of that tertiary structure. They're less folded up and tend to be longer, more elongated. They tend to be more structural in nature. So collagen is a protein in our skin, in our connective tissue, that helps to hold us together, which makes it structural. And then, of course, spider silk is an amazing structural protein that holds together spider webs. So denaturation is this thing that can happen to proteins when we mess up how they fold. So the folding, that three-dimensional structure, which comes from secondary alpha helix and beta pleated sheet, that folding, little bits of folding, three-dimensional structure is more folded, and then quaternary structure. Three-dimensional comes from all of these, secondary, tertiary, and quaternary combined. If we do something that messes up the shape of that folding, we can have what's called denaturation, which is basically changing the shape of the protein, and if the protein doesn't have its shape, it cannot do its job anymore. Sometimes we can renature the protein, put it back into its right folded up shape, and then it can go back to its job, but that doesn't always happen. Sometimes denaturation is permanent, and that protein is dead forever. Some ways that we can denature proteins, and we'll talk about this more when we get to our enzyme slides. If we heat it up like egg white protein, when you cook eggs, that protein goes from being pretty soluble to solid, very, very, very solid. So lots of things we can do to mess up the shapes of proteins. Heat is a big one. And then, of course, if that protein is denatured, we have messed up its shape. If the shape is wrong, it cannot do its job, because protein function is all about shape. So lots of different protein functions, structure, we talked about with those fibrous proteins. Collagen and spider silk are really good examples of fibrous structural proteins. Histones are structural proteins found in our DNA. Hormones like insulin or glucagon, these are both proteins that influence homeostasis. Immunity, we have these guys called immunoglobulins, which are also known as antibodies, that are going to help bind to pathogens so that our immune system can kill them off. Blood clotting, like when you get a cut and you need to scab over that cut, that also is taken care of by proteins. We have transport proteins like hemoglobin. Hemoglobin is going to carry oxygen around the blood on red blood cells. We need that oxygen carried around so we can get it to our mitochondria, so those mitochondria can make lots of ATP for us. Sensory receptors like rhodopsin is going to notice some color stuff in our eyeballs. Movement is a super important function of proteins, so all of our muscle proteins, actin, myosin, titin, are all proteins, fibrous proteins. And then enzymes, which is what we're going to talk about for the second half of today's lecture. Rubisco, catalase, amylase. Basically, if it ends in ase, it's going to be an enzyme, and its job will be metabolism, either catabolism or anabolism. Proteins are also involved in lots of technology, which is super cool. So we put enzymes into our detergents, and these enzymes will digest dirt on our clothes. Diagnostic tests like pregnancy tests, we actually use proteins, those antibodies, immunoglobulins, that we can stick to the stick, and then they will bind to other proteins, and we can use that for diagnosis. Pharmaceuticals like insulin, we can use proteins like insulin to treat diseases and disorders. And then our food, we can use enzymes like lactase to make lactose-free milk for our friends who are lactose intolerant. So now we're going to flip over to this idea of enzymes. Enzymes are special kinds of proteins that are involved in metabolism. So these are our metabolic proteins. And these guys are super cool because they have an active site, and the active site binds to substrates. Super important to know that enzymes have active sites and substrates bind to the active site. The substrate is the thing that the enzyme is going to be acting on. So if we're talking about the enzyme lactase, lactase is going to bind to lactose to break it down into those two monosaccharides, glucose and galactose. That is the job of lactase. So this is the enzyme ACE. If it ends in ACE, it's an enzyme. Lactase is going to break down lactose. Lactose is going to be the substrate. That lactose is going to bind to the active site. So if this is lactose, it's going to bind to the active site of lactase. What's super cool is that they tend to have shapes that are very complementary to each other. So that substrate lactose will bind to the enzyme lactase. Lactase is going to put some stressors on the chemical bonds that hold together those two pieces, and it'll make it easier to break that bond and chunk those pieces apart. So you have two different theories of how this fit between the substrate and the active site works. We used to talk a lot about lock and key, where they literally had complementary shapes that were exact shapes that exactly fit all the time. And you can see on this example how perfectly complementary those shapes are. We think now that a better theory is this idea of induced fit, where there's like a spot that's a fairly good fit, substrate to active site, but it's not until that substrate binds that the enzyme is going to perfectly wrap around and have that matching shape. And so we call this induced fit. The enzyme is induced to fit around the substrate when the two bind together. So that enzyme substrate complex is what triggers that perfect little shape. Little shape. It's a huge, important, big shape. So just one more quick review of how this works. We've got this substrate. Perhaps that substrate is lactose. Lactose is going to bind to the enzyme lactase on its active site. We now have that enzyme substrate complex. The enzyme is going to put some stressors on this bond and make it easier to break the bond. We're also going to add some water because this is a hydrolysis reaction. And then we're going to get glucose and galactose, if this was lactase, or if it was maltose and maltase, we'd get two glucose molecules. If it was sucrase and sucrose, we would of course get glucose and fructose. Enzymes don't just break things down though, enzymes can also build things up. So we can take two little pieces, we can have our monomers, maybe these are two pieces of glucose, and then this enzyme is actually going to build maltose instead of breaking down maltose into two glucose molecules. So now we can make maltose here. What the enzyme does this time is helps to form that bond right there. We're going to take a water molecule out because this is now a condensation reaction. This one is going to be anabolism. And we're going to use a whole lot of ATP to make this happen. Good? Good. All right. So we can change the rate of enzyme activity by changing how these collisions can happen. And that usually depends on the substrate concentration. So the substrate is going to undergo its chemical reaction with the enzyme only if it can get close to and bind to the active site. So if I have like an enzyme over here and my substrate over here, nothing's going to happen. But if I can put these guys into contact more frequently, we have more collisions and we're going to have a faster rate of activity due to the collisions happening between the substrates and the enzymes. Collisions occur randomly because these guys are rolling around each other. And so it's only when we have that random collision between the substrate and the enzyme that reaction is going to happen. If I increase the concentration of the substrate, we're going to have more collisions. And if I have more collisions, we're going to have more reactions. More collisions, more reactions. And so if I have a low, these brackets are our shortcut in chemistry for concentration. So if I have a low concentration of the substrate, low substrate concentration, at the beginning of our graph, we have very little pieces of substrate. We're not going to have a very high rate of enzyme activity. However, as I add more and more and more substrate, more and more pieces, we're going to increase that rate of enzyme activity because there are more collisions. But then at some point, all of our enzymes are bound to substrates. They are working as fast as they can. And so it doesn't matter how many more pieces of substrate I add to the environment, those enzymes are maxed out. So we have said that the enzymes are saturated. They are working just as fast as they can. They are all bound to substrates and they're doing that metabolic work as quickly as they can. They just can't work any faster. We have saturated them, we've maxed out, and we say that this graph has plateaued. Good, good. Temperature can also change the rate of enzyme activity. So if things are very, very cold, when things are cold, there's very little movement. And if we have little movement, there are very few collisions. And if there are very few collisions, the rate of enzyme activity is going to be very, very low. If I start to warm things up, we're making that temperature go up, up, up. The warmer things are, the more movement there is. And if there's more movement, then there are going to be more collisions. And if there are more collisions, we're going to have a higher rate of enzyme activity to a point. And we're going to call that point the optimal temperature. And at that optimal temperature, we're going to have the fastest rate of enzyme activity. For us humans, it's usually just about 40-ish degrees Celsius because human body temperature is 37 degrees Celsius. And so 40-ish, just a little warmer than body temperature, is where our enzymes work the best. When things start to get really, really hot, when things warm up too much, now our enzymes are going to be denatured. All that heat is going to shake apart the folding of the proteins of the enzymes, and if the enzymes lose their shape, they cannot do their job anymore. And so we have a really rapid drop-off in activity. Those enzymes are misshapen, they cannot work anymore. pH is another way that we can denature our enzymes, and the reason is this. We have these super cool hydrogen bonds because oxygen is a little bit negatively charged and hydrogen is a little bit positively charged, and so we have these forces of attraction between the negative oxygen and the positive hydrogen. If I add an acid, I'm adding hydrogen ions which are positively charged, and these hydrogen ions are going to float in and stick to those oxygen atoms. And if the hydrogen is stuck here, then there's nothing left for this hydrogen to be attracted to, and all this folding will just break apart. So the hydrogen ions from the acid will interfere with the hydrogen bonding, which holds together those enzymes. pHs also come at optimal levels. And so we'll have, for this guy, the best pH is 8, and that's where we're going to have the best enzyme activity. If we go away from that optimal pH, we have a quick drop-off in activity because we're changing the shape of the enzyme. What's really cool is that not every enzyme has the same optimal pH. So pepsin's optimal pH is like 3, and trypsin's optimal pH is like 8. Both of these are in the human body. How does that happen? Well, hopefully you're thinking about the fact that pepsin, pH of 3, is probably in the stomach, which is quite acidic, whereas trypsin is found in the small intestine, which is more neutral, just a tiny little bit basic. And so we have different optimal pHs for different enzymes in different parts of our body. And then we have these things called inhibitors. And inhibitors are going to bind to enzymes and shut them down. So if I have no inhibitor present, the substrate can definitely just stick to that active site and things can happen. If I have what's called a competitive inhibitor, the competitive inhibitor has a shape similar to the substrate. And so the inhibitor will bind to where the substrate should have bound, and it's blocking it. It's literally competing for the active site of the enzyme. And if the inhibitor is blocking the active site, the substrate can't get there, nothing happens. Now that competitive inhibitor won't stay there forever. It'll float away, and that competitive inhibitor can come back and bind, and then stuff can happen. We also have the idea of non-competitive inhibitors, and these guys are weird because they don't do anything in terms of binding to the active site. They do not bind to the active site. Instead, they bind someplace else, some other part of that enzyme molecule, not the active site. They bind someplace else. We call that the allosteric site. And you can remember this because in Spanish, allá means over there. So not here on the active site, but allá, over there on the allosteric site. What happens when the non-competitive inhibitor binds to the allosteric site is that we change the shape of the active site. So if we go back here, if we look at the active site here, it looks like this. It looks like this. Now the active site looks like, oh my gosh, it doesn't look like that anymore. So when the non-competitive inhibitors bind to the allosteric site, some molecular changes happen inside the enzyme which changes the shape of the active site. And now there's no way that that substrate can bind to the active site because their shapes don't match anymore. So we can look at the rate of reaction and how different inhibitors will impact that rate of reaction. So if I have my normal enzyme, no inhibitors present, we're going to have that increase in the rate of reaction as we have more and more substrates, we have more and more collisions happening, and then we have that plateau. When I add in my competitive inhibitor, the rate's going to be less, less, less because those competitive inhibitors are blocking the active sites. But if I have a really high concentration gradient of the substrate, if I have a huge number of substrates, then statistically speaking, the substrate is going to bind to the enzyme more often than will the inhibitor, and we're going to have an increased rate of reaction to a point where the inhibitor is binding more than, the substrate is binding more than the inhibitor, and it's like the inhibitor doesn't even exist. We will plateau eventually at the same spot. Non-competitive inhibitor does not look like that. So we're still going to have that reduced rate to get started with, and then we're going to level off at a lower plateau because it doesn't matter if I have a bunch of substrate floating around. If I have changed that active site, there's no way that a lot of substrate can make a difference. All right, so last couple of slides, we are almost done, my friends. So enzymes and technology. It used to be that we had this idea of vitalism. So Pasteur, Louis Pasteur of Swan Neck Flask fame, said that fermentation could happen only if there were cells, that we had to have cells to carry out fermentation. But then in 1897, this other guy, Wuppner, he comes along and he's like, well, actually, him and his brother, they're like, actually, look at us. We can take the yeast, we can mash them up, we can take something out of the yeast, and we can make alcohol out of sucrose. And that is fermentation. So Pasteur was like, fermentation can happen only with living things. And then these guys were like, nope, watch us do it with stuff that we took out of a living thing. And so this cell-free fermentation, we could make lots and lots of alcohol by using just the enzymes from yeast. And this won the Nobel Prize back in 1907. Today, fast forward 100 plus years, we have more than 500 different enzymes that we either take out of living things or we build ourselves in labs. Over 500 different enzymes are used in commercial processes. And here are some of the super cool ways that we can use those enzymes. So most of the time we're going to immobilize our enzymes, and this allows us to control the conditions. We're going to prevent denaturation. We can increase the enzyme concentration if I stick a bunch of the enzyme in one spot. It's easier to separate the products of the reactions from the enzymes at the end if I hold the enzymes together, if I immobilize them, keep them stuck to something. And then I can also reuse those enzymes if I can keep them stuck to something that prevents them from washing away. So we can cross-link enzymes, and then we can make these super cool like beads of enzymes that are all stuck together. We can bond them covalently to something. We can adsorb them with like adhesion. We can put them into little bubbles, so another kind of bead-like structure, or we can trap them in a mesh. So these are just lots of ways where we're holding our enzymes in place. And then we can take our substrate and we can just let the substrate wash over the enzymes, and reactions will happen as the substrate is washing over the enzyme, but the enzymes are stuck there. And so I can wash more, and I can wash more of my substrate over, and we're going to get those chemical reactions happening over and over and over again. And that's exactly how we make lactose-free milk. So we're going to take these super cool beads, and these beads have lactase. Let me use another color. That one doesn't show very well. So we've got lactase stuck to all these beads. Remember that lactase is going to break lactose down into glucose and galactose. And so when we pour the milk through this funnel that has these beads that have lactase, the milk is going to flow past all these beads. And as it flows past the beads, that lactase is going to grab onto the lactose and turn it into glucose and galactose. And so as that milk flows and flows and flows, all of the lactose molecules are going to be converted into glucose and galactose by the lactase that is stuck to those beads. And so by the time the milk gets to the very, very bottom, there's no more lactose in the milk. Instead, we have glucose and galactose. And sometimes lactose-free milk, like Lactaid, will actually taste a little bit sweeter. Sometimes this lactose-free milk is a little bit sweeter because you've got that glucose floating around free instead of lactose binding up that glucose with galactose. All right, my friends, we did it. Objectives completed. We know that proteins are chains of amino acids that are held together by peptide bonds, and we know how to draw a dipeptide. We know that amino acids have different properties. Some of them are hydrophilic and some of them are hydrophobic. Some of them are acidic, some of them are basic, some of them are positively charged, some of them are negatively charged, and those different kinds of amino acids will give our proteins different properties. Proteins have 4 levels of structure. Primary is that amino acid sequence. Secondary is the hydrogen bonding that results in alpha helix and beta pleated sheet. We have tertiary structure, that's 3-dimensional folding that comes from not just hydrogen bonding, but also covalent and ionic bonding. And then our quaternary structure is when we have multiple polypeptide chains that are held together by more hydrogen bonds and ionic bonds and covalent bonds. Proteins can be globular or fibrous. Globular tend to be more metabolic in nature, like our enzymes. Fibrous proteins are more structural, like collagen. Proteins can be denatured, especially by heat, and that just unfolds that important 3-dimensional folding, which is going to break the shape of the protein, which means that it also cannot carry out its function, because we know that the function of a protein is related to its structure. Enzymes are special kinds of proteins that carry out metabolism. They are going to be globular proteins. They have active sites, and the active site is what binds to the substrate. We can change the rate of enzyme. Look at that typo. Rate of enzyme activity can be altered by the substrate concentration, by temperature, by pH, and inhibitors. We have 2 different kinds of inhibitors. We have competitive and non-competitive. Competitive inhibitors bind to the active site. Non-competitive inhibitors bind to an allosteric site, the one that's over there. And then we have lots of cool things that enzymes do for us in technology, like making lactose-free milk. Good work today, my friends.